and in my video. There we go. All right, so today is an opportunity for people to ask questions. It's also an opportunity for me to give you a little bit of a rundown of what to expect on your exam tomorrow. Now, I will share my whiteboard, and I'm going to record all of this. Saves your work and offers latest features. I will use the classic whiteboard. Okay. So over the course of this semester, we have covered our major biological macromolecules. We have and so that was the secret for how to succeed on this exam. I cannot repeat that information any more times. When I was just speaking now, I gave you everything that you possibly need to know, not only about biochemistry, but lottery winnings, success in business, and at any rate, I'm sorry my internet went on the fritz. Okay, so we're going to go back to the whiteboard, and we're going to use the classic whiteboard. Okay, so the stuff that we did not cover on any of our exams is really just the last few days. So what wasn't covered on any of our exams was membranes. Uh, the composition of membranes, protein distribution within membranes, how proteins are found within membranes. Do they span a membrane, or are they integral, or are they peripheral proteins? That's kind of what we're going to explore. And so this was not on exam 1 through 4. And so whenever I think of your final exam is going to be about 60 questions. And I will give you kind of a breakdown and say that from the exam 1 content, there's going to be roughly 11 questions. That's exam 1. Exam 2 content, again, roughly 11 questions. Exam 3 would be around 11 questions. Exam 4, again, 11 questions. And then membranes, roughly 16 questions. So that's a little bit fluid, but that's kind of a rough idea of where things are going to be. There might be some slight variations, but it's not going to be dramatic. When you're looking back at, well, how do I prepare for all of these different things? I would look at the top hat questions, as well as old exams, old exams from what you've taken. The old exams that I posted from previous classes, those would be like lower tier priorities or lower priorities. Um, but this is kind of a rundown. I wanted to give a little bit more emphasis on stuff that you haven't been tested on. Um, and this would be kind of that opportunity. Um, so if I were to like triage it and say, okay, what's the first thing that I'm going to look at? And let me go ahead and change this. I'm going to go ahead and say your primary study materials, top hat questions, old exams, and PowerPoints. What I would do is I would rank them as old exams first, PowerPoint second, and top hat questions third. I would think about taking any of these different things, or sorry, any of these things, either old exam questions or top hat questions, and try to rephrase them. Like if the answer to the top hat question, or sorry, if the answer to the old exam question, or the answer to your exam question was something like competitive inhibition, and the question was, where is there an intersection of the challenged and unchallenged example or protein? Then it'd be like, okay, well, it's competitive, that's along the x-axis. I would try to switch that and say, okay, well, what's a type of inhibition where um, KM doesn't change? Or what about if my intersection's on the y-axis as opposed to the x-axis? I'm sorry, I misspoke there. Competitive would be on the y-axis where your intersection is. Um, just try to take the questions and kind of tweak them, change them. Um, I think that's the most effective way to study for my exams because that's basically what I do. Um, at this point in time, I want to address the top hat questions. So I'm going to add this top hat questions. So I have um, Tuesday questions and Wednesday questions, and then I've got 4 review blocks. 1, 2, 3, and 4. These are due Friday. And then these I just left as because I finished posting them today and I wanted to give you some review materials, but I didn't want to like, you know, overwhelm you. These are due on Sunday, and I know that's after your final. Um, but here's the thing. What I'm going to do is I'm going to migrate all top hat question or top hat scores, let me say it, TH scores into the gradebook, and I'm going to drop lowest 6. So I'm going to drop the lowest 6 of those scores, at least 6. You know, after I look at the grades and look at the overall numbers, might drop 7. But right now I'm leading, I'm going to say I'm dropping at least 6. Um, and so what that does is basically if you know, maybe you forgot to do top hat 1 week or something like that, then you can go back and do these right here to kind of offset that. In the event that you're like, well, I look at, you know, we did 18 top hat days and I've got 13 that are great. Well, then you can just go ahead and not worry about these right here, not worry about these right here. It's your call. But the important takeaway from this is I'm dropping the lowest 6. But like I said, it's at least 6. It might be 7, but what I can kind of definitively say is I am dropping 6. Um, so basically, these right here, these review blocks, you know, they're an option. If you want to look at them, you're more than welcome to. If you have no interest in it and you don't want to use your time studying that, then don't worry about it. Okay, then the next thing is that the exam, like I said, 60 questions, and I would split this as roughly 40 to 45 are straight up multiple choice, and then 15 to 20 of them are multiple answer. Uh, and so that's just something that you can kind of anticipate from basically the breakdown of the exam. Then let's see, you're going to have 120 minutes, 120 minutes. And then provided that you have the ODS paperwork in, I'll provide the extra. I guess that would be, you know, if it's time and a half or double time, I'll do that. The exam is going to be open starting at 12 p.m. tomorrow and will close at 7:30 p.m. So you'll have most of the time, and that block of time will cover or it will overlap with when the university or the registrar's office has our exam scheduled to start, which is scheduled to start at 5 p.m. The block from the registrar's office is 5 until 7:30 p.m. So this covers that. If you want to take it earlier, you're more than welcome to. Um, Now, before I go into kind of an overview, I'd like to open the floor to anyone who has any questions, either about content on the exam, um, something about previous exams. It's totally your call. And I will just sit in silence and drink some water. Okay. Well, then what I'd like to do is I'd like to kind of proceed and give what I think is, my overview of this class. Now, whenever I think of this first semester biochemistry class, one of the things that I think of is the central dogma of molecular biology, which is DNA is transcribed into RNA, which is then translated into a protein. Now, from any other class that you've ever taken, any other biology class that you've ever taken, that this like linear path is not quite that simple. But I just start out with it in that or from that perspective. Then what I do is I think about, okay, well, what have we done with respect to this pathway? And one of the things that I like to look back at is I like to look at DNA and RNA, and I think of sugars. And I think about a DNA molecule and an RNA molecule. And I do a very crude model of it. So here's my sugar, here's my base, and then here is my and whenever I said crude model, I was not joking. Um, so this gives me a very rough idea of what this molecule looks like. Now, the thing that I would recommend looking at here with this model is that I haven't really drawn a DNA molecule or an RNA molecule. I've drawn a nucleic acid. But one of the things that's worth noting is that right here and right here, I don't have a base pair. So I don't have something hydrogen bonding with that. So that's a good place to start and say, okay, well, what are my bases? Okay, well, I could have an A, T, G, or C right here. If I have an A, T, G, or C, and I don't have U, well, then I know that I am talking about a single-stranded ssDNA molecule. Now, the next thing that I know whenever I kind of arrive at that, saying that, okay, this is a DNA molecule, is what does that tell me about my sugar? Well, one thing that it tells me is my sugar will lack a 2' OH because the D in deoxyribonucleic acid is deoxy. So the oxygen is absent. And I know not only is it deoxygenated, but I know exactly where. It's at the 2' position, which now I'll go ahead and draw my ribose a little bit better. This is going to go up to my base. This right here is carbon 1' or atom 1'. This is 2', 3', 4', and 1'. Okay, so those are the atoms of my sugar. And I know that a deoxyribonucleic acid will not have an OH group right here. Okay, so that's one detail that I can, extract from this very crude cartoon model. Another thing that I want to take away from this is this right here. I have a phosphate. Now, I have a phosphate going to a sugar and another sugar. Right, I've got my O, P, I've got a phosphate group here, and then there. Okay, so what this phosphate group here tells me is that this is where my or DNA's negative charge comes from. Okay, so it's that phosphate backbone. So that's one piece of information or another piece of information that I've kind of pulled from that. Now, what I haven't gotten into, but is super important, is, well, this phosphate group and what are the bonds that are here and here respectively. They are phosphodiester bonds. It's a B, not a D. So it's a phosphodiester bond or a phosphodiester linkage that's found between my sugars. And it is linking a 3' carbon to a 5' carbon. Now, I need to make an edit to my model that I had just a second ago. Rather than CH2OH, I've got O, P, O, O minus. So what I have is, let's say that this is an A and this is a C. I have a little bit more information about the structure of my molecule and specifically the primary structure of my molecule. I have PA, PC. I asked a question on the first or second, it was the first exam, I'm pretty sure. Um, Maybe it's the second. How many phosphorus atoms are in this molecule? And the key there was atoms. The key there was the atoms as well as it was asking about phosphorus, not phosphate. Um, and whenever I added that phosphorus, then it kind of changed things because the question gave you a single or sorry a primary sequence of a DNA molecule that was like PACG. Well, here's a phosphorus, but then there's another one between A and C as evidence right here. Then there would be another one between C and G, but there would not be one on the 3' OH group. So essentially, the answer to this was, well, there's 1, 2, 3. There's 3 phosphorus atoms. Now, this is a lot to unpack from just a simple DNA molecule, but these are all sorts of questions that I really like to ask because they go into the detail and the minutia of what type of bonds, but then they keep in perspective the big picture of what does this molecule look like? What type of bonding can it do? And then like what's the overall charge of this molecule? So this right here, you know, this is a good sort of exercise for you to think about, like looking at a DNA molecule and basically ripping it apart. What type of structure do I have here? What do I have here? Um, and that's going to be valuable because these are all like I can almost guarantee you this will give like 2 to 3 questions on your exam from this sort of exercise because I'm asking types of bonds, I'm asking about overall charge, and then I'm asking about the details of some of those bonds. And In addition to that, distinguishing between a DNA and an RNA molecule. Now, does anyone have any questions on that? So that kind of captures some of the high points of DNA and RNA, distinguishing the differences between DNA and RNA. But then if we go on to proteins, well, with proteins, there's a lot of information because when you think about primary structure of a DNA molecule, we go into that. But then when you think about primary structure of a protein, it's like, okay, the primary structure is neat, but what about the secondary? Okay, that's cool. What about the tertiary? Okay, that's cool. What about the quaternary? So we go into all these different levels and we go into depth on all these different levels. So protein, we talk about structure. And within that, we've got primary, secondary, tertiary, quaternary structure. Then we've got function. And we use for function, we use hemoglobin and myoglobin as our models. We have these two proteins that essentially do the same thing in a non-catalytic way. They bind oxygen and transport it. Despite these similarities, these two proteins have very distinct differences. For example, hemoglobin will hemoglobin is an allosteric protein. Hemoglobin as an allosteric protein binds O2 and changes its shape in response to that. So when it binds O2, then it says, okay, now I'm going to move like this. Myoglobin, by contrast, does not. That's very significant that when hemoglobin binds O2, it changes shape because when it binds a first O2 molecule, then all of a sudden its affinity for the second, third, and fourth O2 increases. That is a phenomenon known as positive cooperativity. I used an analogy that I really like, uh, that I always think of, and that's like a junior high dance or at a wedding or something like that, a wedding reception where nobody wants to get on the dance floor. That's hemoglobin not wanting to bind to O2. Then finally, hemoglobin binds to that first O2. It undergoes a conformational change. And whenever it undergoes that conformational change, now its affinity for the second O2 goes up. So when it binds the second O2, now all of a sudden its affinity for the third and subsequently the fourth O2 is going up as well. So this is a very strange concept to think about. But essentially what that means is that hemoglobin's affinity for the first O2 that it binds is lower than its affinity for the fourth O2. In other words, hemoglobin would rather bind the fourth oxygen than the first oxygen. Now, as a little bit of a parallel, I always think of this, it was I'm gonna not completely butcher it, uh, but if I remember the quote correctly, it was basketball legend Kobe Bryant said that he would go 0 for 21 before he went 0 for 19 with an assist or something like that. And I think that the idea was that he would rather take all of his shots because he was certain that he was the best player on the field or on the court to make that shot before he would pass it to someone else. So it's not a completely direct parallel, but I think the idea is the same, that hemoglobin would rather bind the fourth O2 that it binds than it would bind the first one. And that's only because the shape of hemoglobin when it's binding that fourth O2 molecule is radically different. So hemoglobin with three oxygens bound has a different or has adopted a different shape than when it's not bound to any oxygens. Now, that deoxygenated form of hemoglobin, deoxy HB, well, that is commonly known as the T state. When we talk about allosteric proteins, we talk about proteins being the T state and the R state. Um, and your T state is your basically your inactive form of the protein, whereas your R state is your active form of the protein. And the same goes for enzymes. It's the T state of the enzyme or the R state of the enzyme. I've heard many times these referred to as the tense and the relaxed state. The relaxed state is, oh, it's ready to bind a substrate and carry on the reaction. Whereas the tense state is a little bit more like it's clammy, it's rigid, it's not going to want to bind that substrate very well. Okay, so we talk about function, we talk about all of the different levels of protein structure. One of the important things, or what's really important about protein structure, is not only to understand kind of conceptually what it is, but the forces that stabilize it. So I'm going to write stab force. Primary structure is peptide bonds, which a peptide bond is an example of a covalent bond. Secondary structure is H bonds from the backbone. This is going to lead to alpha helices and beta sheets. And with these alpha helices and beta sheets and these backbone hydrogen bonds, what we've got is a protein that is basically able to, or we've got interactions between the N and C termini of different parts of the protein. Uh, these form pretty unique patterns, and although this is all backbone driven, there are amino acid R groups that are more favorable or less favorable for these two different structures. Now, when it comes to alpha helices and beta sheets, the R groups or the amino acids that are your backbone or your secondary structure breakers are proline and glycine. And those are just those are good starting points. And it's I think helpful to think about those two amino acids and say, okay, well, what makes them unique? Well, proline is super rigid and glycine is super flexible, entirely too flexible. Um, but I think that yeah, that's helpful to understand that. One of the things that you should absolutely know about the secondary structure stuff is the math. So one rotation that has a height of 5.4 angstroms, and it takes about 3.6 amino acids to complete that one total rotation. So essentially what that means is you could kind of think about the height of an alpha helix or the height of 3.6 amino acids that have formed an alpha helix would be 5.4 angstroms. So if I said something like you have an alpha helix that is 120 angstroms tall, you could go a couple of different directions. So 120 angstroms, that is going to require, just a second, I got to use my phone here. Know that somebody's probably already done this calculation. 120 divided by 5.4, that would be about 22.2 rotations. And if each one of those rotations has about 3.6 amino acids. Then we're talking 22.2222222 times 3.6. So this is roughly 80 amino acids are required to make that structure. Um, so be able to do those sorts of calculations because they're going to be a part of, or they're that's going to be a sort of question on here. Tertiary structure are group interactions. Uh, Now, this is where, to me, protein structure is really interesting because when your R group interactions, or when you talk about your R group interactions, you get to, you get to talk about, well, what does this amino acid do? Well, it could form an ionic interaction. And then that to me is like, okay, well, what amino acid could possibly form an ionic interaction if it's the R group? Alanine? No. Valine? No. Leucine? No. Asparagine? Closer, because you got a polar residue, but more than likely asparagine and glutamine, those are going to do hydrogen bonding. So then when you finally get to like aspartic acid, okay, aspartic acid is a polar residue and it's negatively charged at a pH of 7. Um, Histidine, that can be positively charged. So I always like to start my conversations or my thoughts about R group interactions and basically go to all of my non-polar residues. Will they have R group interactions? They absolutely will. What will those R group interactions be mediated by? The hydrophobic effect. Uh, this is where you kind of hit pay dirt of you know your amino acid structures and you know the types of interactions they can have. Um, I will ask questions about amino acids specifically, like what's the 3-letter code for tryptophan, TRP? What's the 1-letter code for tyrosine? Why? What is the amino acid that corresponds, and what's the full name for the amino acid that is represented by H? Histidine. Um, but then you're going to use that to apply that information when I ask about R group interactions and types of interactions that there will be. That also sets you up for a good understanding of enzymes and catalysis, and to say like, okay, what amino acid can possibly um, participate in, sorry, what amino acids are capable of attracting a divalent cation, as we saw with carbonic anhydrase. Well, chances are that amino acid that's going to attract that divalent cation, it's probably going to have some lone pairs of electrons. Okay, which amino acids could have lone pairs of electrons? Well, asparagine could, glutamine could, um, lysine could potentially, um, aspartic acid could. It just depends. Like that's where you're going to benefit from knowing how your amino acids ionize and what they look like and how they differ from one another. So this is like understanding protein structure and understanding amino acid structure. Those two things go hand in glove, and they kind of help one another. Your quaternary structure is R group interactions, but your difference is this is one peptide and this is two plus peptides. So all worthwhile to think about. The last little bit or the last portion of your exam is lipids. Um, yeah, it's lipids and membranes. Um, I will tell you that I really, 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 really love that table from our lecture on E. coli grown at different temperatures. So I would definitely make sure that I understand that because I'm probably going to have a question or two based on that. These are, to me, these are like the major high points. Um, and I think that I'll just I'll open the floor for any questions, but before I do, I'm going to go ahead and stop my recording.